Hello, my name is Carla Mendez Muñoz. This year I went to Simvestab Monterrey to make my professional practices. The name of my project was Electroencephalographic Signal Analysis. The base of this project was the acquisition and processing of electroencephalographic signals. First, we obtained a 3D model of the skull, and then we compared two types of electrodes by comparing two types of caps. The first, ECI cap, which uses silver electrodes, and the second, Movita cap, which uses water-based electrodes. We measured the signal in 6 specific channels and obtained a specific signal called the P300. The main objective was first to implement the Polemus software, which was used to create the 3D model, then learn how to use both Movita and ECI caps to obtain the P300 signal, which was already known and already studied. This signal was used to compare both caps. As it is already known, we can know what is going to happen and how it has to look, so we can compare the quality of each one of the electrodes and to see whether it's better to use water-based electrodes or the common used silver electrodes. The goals that come along with these project objectives are the following. First, we had to learn how to digitize head points using MATLAB and Brainstorm, using the Polemus Fast Track software. This one to make sure that we were taking good digitalization points. Apart from this one, we had to learn how to use the caps. First, how to put them on, then the positions of them. There are positions on the caps that have names, specific names, and we only needed to use 6 of them. So we had to study the map of the placement holes. Then we had to learn how to use water-based electrodes, which is very simple, but still we had to do it because we had never seen anything like that. Also, we had to learn how to measure impedances for silver and silver chloride electrodes, which was a little bit hard, but it was okay. Then to process the signals to get the P300 signal, this one with the help of MATLAB. And then note and compare the obtained signals to compare the two types of electrodes and decide whether the water-based electrodes are good or the normal silver ones. To accomplish this, we had to learn how to use both caps. First, the ECI, which is the red one, then the Movita one, which is the gray one. For both cases, we had to learn how to do some measures so that they are placed in the correct place. So, for the first measures, we had to locate the nation and the union point of the head. So, measure the nation to union distance with a tape measure. Then take the 10% of that distance and measure it from the nation up to the forehead. Then measure the circumference of the head, making sure that you place the tape measure horizontally on the forehead. Then measure 5% of this distance into one side of the previous mark. We have a mark here, 5% to the right, 5% to the left. Then, with the first one, the ECI, we have to put 2 sponges, one on the point on the left side, which is FP1, and one on the right side, which is FP2. This is used so that the cap doesn't move from its place. Then we have to put the cap on to the final part of the head. Later we have to place the reference, which is placed on the earlobe. First we have to subject to clean the earlobe with a little bit of alcohol. Then we put some conductive gel on the reference, and then we place the clip on the earlobe. Then we have to measure the impedance of this one. As we have to put both earlobes, we can have two electrodes on the device and we can measure their impedance. This impedance must be lower than 5 ohms so that we can know that there are not going to be a lot of interferences. Then we have to put the straps on the cap on the harness that we have previously put. Then we have to put the conductive gel onto the electrode on each of the points that we are going to use, and we have to measure the impedance of these points. If the impedance is too big, we have to, with the syringe that we are injecting the conductive gel, we have to do some circular movements so that we can put away some of the hair that is blocking the conductivity of this experiment. And we have to make that all the points that we are going to measure are lower than 5 ohms. Then we can start with the acquiring of the signal. We have to connect the electrodes to the adapter to put on the Movita device. Now I'm going to explain how to put the Movita cap. First, we have to roll up the water-based paper electrodes. Then we have to put it in the electrode. Then the electrodes that we're going to use have to be soaked in water, plain water, for a few minutes while we put on the cap. On the previously marked points, we have to place FP1 and FP2. We have to put the cap onto the head, and then we have to pull away some of the hair that's on the holes. We use the stick and our fingers to move the hair so that there is no interference. We have to see a little bit of the scalp and the skin so that we can know that it's going to be a better signal. We cannot measure the impedance from these caps, so we just put, after again putting away the hair, we put on the electrodes already soaked in water. The subject must feel that it's a little wet, and then we connect the device to the Movita device. We connect the upper part where the electrodes are connected to the bigger part of the box, which is the Movita device itself. It's important to mention that the Movita device is a device that can be connected wirelessly to the computer and uploads the information to a program called Acknowledge. This program, we have to do a configuration that we can see on the right side of the screen. There is this window we have to configure it. In the analog tab, we can see that we have activated 8 channels. The first 6 for the 6 channels that we had mentioned before, which are on the central part of the head. These ones are electroencephalograms, and the final 2 are electrocardiogram. These ones are very, very important because when we have already acquired the signal, we have a lot of artifacts that make noise in the signal, that makes the signal be less quality. So for these electrocardiograms, we have to put an electrode on the upper part of the eyebrow, exactly on the center of the eye, and the other one on the cheekbone, exactly on the center of the eye. So in the program Acknowledge, we use the calculation tab to do the subtracting of the upper one minus the lower one. So we can have the complete electrocardiogram signal. And then we have to, when it comes to the processing part after the filtering, we have to remove the artifacts by subtracting all of the signals minus this electroencephalogram signal. Later we had to do all the processing, which was taking the exact moment when the target sound was presented. We did this by the trigger. With the trigger graph, we obtained the exact point when the stimulus was made. So we took that point and go backwards 500 samples, and then from that 500 samples before to 1,500 later, we obtain a graph of that. So in the sample number 500, there was the exact time where the target was presented. So 300 milliseconds later, we had to see the P300 on the graph, which was on the sample number 800. As I'm going to explain later, the graph had to have an amplitude, a greater amplitude in the P300 signal. Then we averaged all the targets of the experiment, which were about 60, and we made an averaged graph of each channel. So we had at last 6 channels and 6 graphs of an average signal. This one we uploaded into the Polemos digitalization, and we could see all the signals on the scalp on a 3D way. So we could see all the channels at the same time, and we could see the configuration of the signal, of the energy, whether the energy was on the frontal part, on the central part, on the back part of the skull. And the thing that we expected was that it was on the central and the frontal part of the brain, as we are going to see on the next part of this presentation. As our experiments were not invasive and not really complex, we did not use any regulations to apply them. It's important, however, to mention that the Movita device is not intended for medical use, but in the case that it was, it would have to meet some regulations. I found this regulation of the FDA, which is used for neurological diagnostic devices, and says all the regulations that it must have in order to compete on the market. The contributions that we made for this project was that we learned how to use Polemos Fast Track software to implement the 3D model skulls. So as we can see in these pictures, we managed to do it very well. Also, we learned how to import EEG signals in this software to see the signal on the scalp. As we can see in both results, this energy of the P300 was concentrated on the central and frontal part of the brain. Both of the images are taken of the channel 3 and on the greater point of the Polemos of the P300 signal. The other contribution was they learn how to use the water-based electrodes and to compare if it was better to use water-based or silver electrodes. We learned that both caps are really good, that Movita cap could be a really good option when it comes to time because it's really easy to use, really fast to put on, and the signals acquired are not that bad. We do think that there was a problem with the size of the cap because it was a little bit loose in some parts, and we believe that that's why we had a lot of interference. Because as you can see here in these pictures, the first one is of ECI cap, the second one is Movita. The first one has a very beautiful P300 curve, while the second one has the curve but it's a little bit noisy. So this is the main thing that we discovered, that water-based electrodes are good, but the silver electrodes are still better. But we hope that we or they can investigate more about the size of the cup, because maybe they can make it tighter and make the signal be better. In conclusion, water electrodes are a useful and easy to operate tool. However, we still believe that silver electrodes are better to use because of the noise present on the water-based ones. We also learned that the signal is mostly concentrated on the central part of the brain and the frontal part. We hope that this investigation that we did can help further investigations, and that the students can use this information, because there are infinite possibilities when it comes to the brain, and the better and the more we know, it's the better. It's important to say that we did everything with ethics, and that we respected the IEEE Code of Ethics. Thank you very much.